kind of what they are up to. I took one poll. I think there are a couple of more that I need to. And communication over these different channels has been interesting. Like you can click on what you're interested in and find out what's happening, ask more questions, answers. So.
Yes. So we're about 2 minutes out by my clock. Oh. So Nikki tells me that there's a bug in the poll on acronyms because you can't submit an empty response.
I don't, Nikki, what do you mean? I had no trouble leaving, an empty response.
Empty response, okay. That she says, or they say that I thought we should explain all terms for maximum accessibility. So, and I agree, yeah.
Yes, I had the same issue. Yeah.
I see.
Ah, well, I should. Yeah, it stopped me at 10, and so yeah, I was kind of hoping there would be something where I could do none of the above or all of the above or none of the above actually, or an other, and but that didn't work. But yeah, that's a good point. Well, I'll have to see what I can do. Maybe close that poll and start another one that includes a none of the above. So Marcus should be here. Can we let him in and it's and make him a co-host, please? And I think we're going to go ahead and get started. Hello, welcome to TDWG 2021 and the virtual conference. If you have not already been welcomed today, this is the contributed oral session, the first one, and I am your moderator. My name is Gail Kampmeier. I'm in Illinois in the United States, and my co-moderator is Terry Young, and she's in Paris. And we're very grateful for the tech support from the University of Florida's conference team. This session will be recorded for later on-demand viewing, and we thank you very much for the speakers in the session. You probably know that each presenter will be allowed 10 minutes to talk. There will be 3 minutes for questions at the end of each presentation and 2 minutes to transition between presenters. Please speak slowly and clearly for our international audience. Note that in this session, 2 of the presentations are pre-recorded and 1 is in person and 1 has been canceled. So it'll be a little shorter than we first expected. Please ask questions of the speakers in the Q&A feature in Whova, and these will be asked by the presenters by the co-moderator, Terry. Please also specify whom you're addressing the question to, as the Q&A covers the entire session. There are 2 chats available, one in Whova and one on the Zoom screen. The one on the Zoom screen is not persistent. So if you have information that you're sharing that other people should see, please use the one in Whova, because this platform will be available for quite a long time, certainly over the week, and others can benefit from the links that you share. You can, when this becomes an on-demand feature, then you can still use the Q&A and the chat. But please use the chat, or please use these judiciously, as any inappropriate use of the chat may result in you being removed from the session or the chat function being disabled. We know how to kill specific entries, I think. Please see our code of conduct for more information. And bear in mind that any technical difficulties we may have are due to the Zoom gods or somewhere else, and enjoy the session, please. So first up is a recorded presentation from Robin Boyd, Practical Considerations for Implementing Species Distribution Essential Biodiversity Variables.
Hi there, my name's Rob Boyd. I am a quantitative ecologist at the UK Centre for Ecology and Hydrology, and I'm going to talk about practical considerations for implementing species distribution essential biodiversity variables. So this work was born out of discussions with various colleagues: Nick Isaac, Rob Cook, Francesca Mancini, Tom August, Gary Powley, Mark Lobby, and David Roy. So firstly, what are species distribution essential biodiversity variables? They're a middle ground between raw species occurrence data and high-level summaries of biodiversity change, things like distribution indicators. And the general concept of a species distribution essential biodiversity variable, SDEBV, is a species space time cube. So the idea is to characterize species distributions, so that could be occurrence or probability of occurrence along the axes of space, time, and taxonomy. Various authors have conceptualized what they call the ideal or the idealized SDEBV. Yates et al. 2019 are one of these sets of authors, but there are others. Kisling et al. 2018, for example. And these authors make several stipulations that these SDEBVs should satisfy. But I'm just going to focus on those of Yates et al. 2019 in this talk. So these authors suggest that the ideal SDEBV should populate the species space time cube contiguously in space and time, so populate every cell. They should span the global extent of the focal species geographical ranges, and they should span all members of a taxonomically or ecologically defined group. Now, there are some implications of these stipulations. Basically, in suggesting these ideal criteria, these authors are making one of two sets of assumptions. Either we have structured monitoring data for all species of interest across the entire geographic ranges in regular spatial temporal units, or we don't have this type of data, but we do have statistical models that are able to take information from the cells that we do have data for and extrapolate that information into cells for which we don't have data. So how realistic are these two assumptions? Do we have the data? The answer is quite simply no. I'm not going to go into the specifics of this because I could talk about particular taxa, parts of geographic space, time periods, and I don't have the time to do that. Instead, I'm going to point you to some literature. So these are just 3 examples. There's many, many more papers that show that there are huge gaps in terms of where has been sampled, when has been sampled, and what taxa been sampled. So we don't have the data to populate the species space time cube contiguously across all the geographic range of the species of interest, etc., etc. Unless we're talking about some very specific cases where lots of effort has been expended to collect this sort of data. Generally speaking, it doesn't exist. So The next assumption then, and this must be the assumption now, given that we don't have the data, is that we can use statistical models to extrapolate from the portions of the species space-time cube that we do have data for into the portions that we don't have data. And there's lots of different types of models that you can use to do this, and I've split these models up here into those types of models that are well suited to populating the spatial dimension of the cube, those that are well suited to populating the temporal dimension of the cube, and some models that are in the middle and kind of in principle do both. So to populate the spatial dimension of the species space-time cube, people typically go to static species distribution models, establish relationships between environmental covariates and species occurrences. And this means that they can extrapolate those predictions across the entire geographic space for which they have environmental data, which is typically the entirety of geographical space, can be global. But the problem with these models is that if you want to populate the temporal dimension, you need to use space for time substitution, i.e. you need to project your models onto new covariate data, which has been shown to be quite ineffective, or you need to fit several models to different temporal subsets of the data, which is quite an inefficient way to do things. And it substantially reduces your sample size for each time period, of course. The other side of the coin are what I'm calling temporally explicit distribution models. There's things like site occupancy models, dynamic occupancy models, reporting rate models. Any models basically in which the parameters denote temporal changes in species distributions or range sizes. Now, like I said, these models they're reasonably well suited in the right circumstances to predicting temporal change in species distributions, but they're not very well suited to predicting spatial variation in species probability of occurrence, for example. They're typically not used to produce spatially explicit outputs. So again, in this case, you populate one dimension, the temporal dimension, but not the entirety of the spatial dimension. So it's the other side of the coin of the static SDMs. In the middle, you've got what I'm calling spatio-temporal models. So this would be an extension of your temporally explicit models and your static SDMs. It would be sort of a combination of the two, where you include environmental or spatial covariates, but you also include parameters that denote changes in species distributions over time. In principle, these models can populate the species space-time cube in its entirety, but given the scarcity of data for most taxa in most parts of the world and for most times, there won't be sufficient information there to estimate the many parameters that these models contain. So you're just going to end up with lots of parameter uncertainty, basically, unless again you're in these very specific circumstances where there's lots of data, in which case spatial temporal models might well be the way to go. Now, just to add to this mix of problems, you also have biases in species occurrence data. So I'll show you some literature on the previous slide. It's basically suggesting there's lots of gaps in space, time, and taxonomy. And what this means is, in many cases, the occurrence data sets are telling us more about the distribution of sampling than the distribution of the species themselves. And this can be a real challenge for models, in that the models often cannot overcome these biases. There are various ways that people have tried to do it, so using pseudo absences, the target group approach, factor bias out in Maxent for species distribution models, or using visit level covariates in dynamic occupancy models that give some indication of the distribution of sampling efforts. But there's lots of problems with these methods, basically, and it's also very challenging to understand the extent to which they have helped the models to overcome the biases, unless you have a separate random sample that you can compare the outputs to, which is almost never available. Otherwise, we just use that for the model. So even if we can fully populate the species space-time cube, we don't know whether and to what extent it's biased. So probably been rather negative for the majority of this talk. So I'm going to try and end on a slightly more positive note and discuss what we can do about this. Well, firstly, we can clearly assess the spatial, temporal, and taxonomic extents of the available data, as well as its representativeness of those extents. In other words, you might have data for the whole of the UK, but do they resemble a random distribution of the UK? Has it been a random distribution of sampling, or has it all come from the southeast? And these assessments will basically tell us what we can and what we cannot talk about. It's better to acknowledge that the ideal SD-EBV might not be possible than to pretend otherwise, than to use a black box statistical model and fully populate the species space time cube and act as though this is a robust piece of information that can be used to derive indicators and inform policy. Thanks very much.
So that was the end of the recording for Rob Boyd. Are there any questions that we want to put in the Q&A for him to answer later?
Actually, there's no question yet.
Okay. So between this morning's talks by the plenary speakers and the discussion afterwards, the gap between policy and the data that we have, and knowing the biases, and the policymakers not really realizing perhaps or ignoring what these biases are, is there something that this community can do to help that? I haven't written out that question. This is out to anybody who would like to answer. Please just unmute yourself. Too big a question, I guess, or too, anyway. Shall we go on to the next one? And this will put us back on time. Our next speaker is Sarah Hansen, and she'll be speaking about building the Digital Extended Specimen, a case study of the invasive European frogbit, Hydrocharis morsus-ranae. Linnaeus. Take it away.
So yeah, my name is Sarah Hansen. I am a graduate student at Central Michigan University. My collaborators on this talk are Anna Monfils, Jutta Buschbom, and Karim. Sorry, slide doesn't want to change. Okay. The extended specimen was first defined by Webster in 2017 as a constellation of specimen and data types that jointly add research value to each other. There are many ongoing global conversations about the definition and implementation of extended specimens or digital extended specimens. And these are just some of the groups leading those discussions. I know many here are familiar with these and probably involved in one or many of these. We'd like to frame our perspective in our use of DES concepts with a real-world example of European frogbit, which we will present here as a case study. European frogbit, or EFB, is a free-floating aquatic plant invasive to North America and India, and native throughout much of Europe and Western Asia. We first saw this in North America as an ornamental in 1932. Since then, it has spread throughout Ontario and Quebec, Canada, and the northeastern and midwestern United States. The first observations of EFB in India were in 2013, where it has since had considerable impact on ecosystems and subsistence farming. In both North America and India, researchers and managers are interested in understanding EFB's pathways of spread, its effects on ecosystems and biodiversity, and the potential for eradication, effective control, and methods to mitigate socioeconomic and ecosystem impacts. We've been working with national and international partners to build and continually update a comprehensive data set of EFB occurrences across the invasive range of North America. We aggregated over 23,000 records dating from 1932 to 2021, And after our data curation efforts, have 12,284 records of EFB spanning 13 US states and 2 Canadian provinces. These data were contributed by over 150 original data providers, including individual researchers, herbarium collections, and state and federal agencies conducting monitoring, among others. These data came in the form of published observations on online aggregators, observations on community-driven science applications, targeted research conducted by our lab and our collaborators, and digital specimens that are either published or were shared with us individually. In the process of integrating these data, we encountered a wide range of different data cultures and how disciplines and communities view data accessibility, data standards, and data sharing. Taking a step back, as we're doing this work, we're following along with these conversations about the digital extended specimen regarding data standards, fair data, and how we can make our own data set accessible and shareable long-term. So we first had to think about how each of our data points could actually fit into the DES framework. All the occurrences within our data set are represented by what we'll call tokens. We're using the word token as described by Boskoff and Webb 2016 to mean a representation of the occurrence of an organism in time and space. So physical specimens within collections are one type of token. In our case, we would be collecting an individual EFB, maybe with its ramets, creating a specimen sheet and label, and adding it to our collection, as well as digitizing it. An observation record is another type of token that we would collect in the field by immediately recording it to iNaturalist. Because these are real time and have images attached, we've actually been able to use them to be alerted to new occurrences in Michigan. We could derive tokens from literature like we did for a single record of EFB in New Jersey where no other record types were available. We could also take pictures in the field to show EFB in its habitat and sample the DNA either in the field or from the specimen if we want to answer specific research questions. Traditional and local knowledge can be translated to tokens. In many cases, the areas we're collecting specimens are managed to some extent by tribes throughout Michigan. The Saginaw Chippewa Indian tribe, for example, has been monitoring and managing some of our sites and has information about these populations that we can directly link back to each individual specimen that would have been directly affected by the management. Other types of tokens we may consider for EFB or other organisms include audio files, tissue samples, environmental DNA, and even anecdotes passed on informally. Each of these tokens represents a different type of information we have about one individual organism. Tokens vary widely in their quality and information content across a set of metrics. I'll share just a few examples of what we are interested in that create different token information content and quality. And these are all continuums, so there's really no way to assign good or bad quality based on any of these. We look at the persistence of a token, all the way from one that's not mobilized in any way, such as our own lab data, to a fair digital object, or something like a digital specimen with a linked iNaturalist record. We also look at the identification, our confidence in the identification of the organism. For example, an identification by someone who has never encountered it before on the low end, all the way up to identification by multiple people with a lot of experience with EFB on the high end. We look at resolution, all the way from no location in the case of some historical specimens we've downloaded, up to a specimen we've collected ourselves with precise coordinates taken with a GPS unit at the time of the occurrence. The metrics we choose to consider and our threshold for what is high enough quality depend on many factors and are going to be biased because we're looking at our experience, the question we're trying to answer, the project, the availability of the data, and many other factors. We can create multiple tokens from a single occurrence of an organism in time and space. From one organism, we could create an observation record, a specimen, DNA sequence, a tissue sample, take photos in the field, and link data from local tribes involved in management. I'd like to go through the process of actually building a digital extended specimen from an individual EFB. So we start with the living organism and decide the two tokens we would like to make are an iNaturalist observation and a specimen. We create the iNaturalist record right away, then pull the EFB out and make a specimen from it. Both of these could get independently published and we could actually stop there. Or we could link these records together using the existing infrastructure of identifiers in our database. Our collector always notes which specimens were made from which observations of individuals. So once both of those are out there, they're easy to link together. Now imagine if we see two separate EFB individuals. From both of these, we could create observation and specimen tokens and link them as I just explained so that we have two separate records of separate individuals that are linked within themselves. These separate tokens from separate individuals may be linked to one another across time and space as well. I'll talk about just one token type for a minute here, a specimen, and go through the process of how these would actually be linked. So say we go out to Fish Point, which is the red star in Saginaw Bay, Michigan. We spot one EFB individual and create a specimen from it. If we look at time on the x-axis and space on the y-axis, we see that that specimen is tied to a particular time and space of that EFB occurrence. Now say I actually see 5 EFB individuals in roughly that same area of Fish Point on that same day. I would say that is one sampling event and create 5 unique specimen tokens from 5 unique occurrences. If I were to return to Fish Point again on a different day that week, I would have another separate sampling event. Because these both occurred in the same location, but at a different time, I would consider these 2 sampling events to be part of one project, which would, in this case, is sampling in Fish Point over the course of 1 week. Maybe I decide to go to another location on the same day as one of my Fish Point sampling events, this time Buschbom Bay, which is the northern red star. I again collect 5 unique specimens from 5 unique EFB individuals. Now I want to call all the events from that day in either Fish Point or Buschbom Bay my project. Whatever I decide will be the time and spatial scale of events and projects, I can link tokens of individual occurrences within and across them. So if I have 2 records from 1 day of sampling in one location, say this goes back to those 2 individual living EFB specimens, I would link them to indicate they come from the same event. Because our sampling takes place over the course of an entire summer, we end up with a lot of individual sampling events. If they all occurred in roughly the same space or roughly the same time, I would link them to indicate they come from the same project. At this point, we have a project made up of only EFB sampling events, but we might have data from other taxa as well, which adds complexity at the ecosystem level as we're sampling beyond EFB. These taxa all have their own observation records or specimen records, or even these linked specimen observation records like we have for EFB. We can integrate all of these into a single database as long as we pay attention to the ways each token is linked to all the others. By doing this, we're actually making extensions at all of these levels, and we could go much further if we want to. We could link, for example, to other projects of the same taxa, other projects within the same location, or specimen or observation records from outside sources beyond our own project. As we conceptualize and build out these extended specimens within just our own database, we might encounter a few challenges. Integrating data from 150 providers means we could have 150 different data standards, token information content, and quality across our chosen metrics. Once we do mobilize these data and make them available for continued extensions, we have to consider at what point each token is being extended and how we could track those and get them back into our own database. Despite the challenges we face, the practical application of the digital extended specimen can open avenues for more integrative research that is scale independent. If we're able to integrate and mobilize data from all token types, we'll end up with a more powerful data set that allows us to look at mechanisms and pathways of invasive spread, create predictive models to determine areas of highest impact or highest concern, and incorporate environmental and climate data to project long-term socioeconomic and ecosystem effects of invasion across time and space. By extending each specimen and extending our data set, we could explore these questions at the global, regional, and local levels. I'd like to acknowledge all those who helped us aggregate our data set, as well as the many data providers and aggregators. Our work with this data set was funded by the Great Lakes Restoration Initiative through the U.S. Fish and Wildlife Service. I'd also like to acknowledge once again the many groups involved in discussions about digital extended specimens. Thank you all very much for listening. I'm happy to take your questions.
Thank you very much. I see we have a question. Terry, you want to?
Yeah. So the question is from James. He's asking, do you use a standardized vocabulary to define your links?
We do primarily use Darwin Core vocabulary when we can. And then if in certain situations, if we need to sort of create our own standard vocabulary, we do that as well.
Okay. Another one from David Shorthouse. He's saying that it's an interesting abstraction of the lines of evidence as tokens. Have you, have you grappled with what is an occurrence, especially when an individual is both observed and collected?
Yeah, that's one thing that we definitely, I think that's why we sort of make the distinction between tokens versus occurrences, observations, specimens, all those. Because you could certainly have many different tokens from one occurrence. And that would be a situation where you would really want those to stay linked together so that we don't end up with looking like more occurrences than we actually have.
Okay, thank you. Now, another one from Nikki. The recent GBIF governing board meeting had a presentation on the redevelopment of Darwin Core to better model field sampling. Sorry, just. Your overview of the field collection sounds like it fits well with that. Are you contributing to the modeling discussion?
We are not at this point, but that would be an interesting thing to look into for sure.
Thank you.
So Nikki provides a link that you can go back to and find out more and perhaps join this discussion.
Thank you.
Debbie's raising her hand.
I would just say, is GBIF in the house? I think, is Tim here? I'm not sure. I would just add to loop you in, Sarah, this recent GBIF meeting that Nikki's talking to. Nikki, we did reach out and ask if they wanted to share that more widely with everybody, and there's a plan going forward to do just exactly that, and to reach out and do some webinars and some more checking with the community on the modeling. So it'll be an opportunity for you, Sarah, for sure. Thanks, Nikki, for bringing that into the conversation.
Yeah, thank you both.
There's another comment questions from Erica. So she's saying that it sounds like one of the products of your work is an enhanced data sets worth being shared. Have you figured out a good mechanism for sharing this data, given that many of the records are already published as e.g. JB specimen records or iNaturalist observations?
Yeah, I think that's one thing that we're working toward, and kind of part of following along these conversations is making sure that we're doing that in a way that is persistent and we're not ending up with a lot of duplicated records all across all these different projects. Definitely something we're working on.
Are there?
Thanks. That's all for now.
Okay, and that wraps it up. Thank you very much, Sarah.
Thank you.
The next presentation is also recorded by Marcus de Almeida. Digitizing primary data on biodiversity to protect natural history collections against catastrophic events. The type material of dragonflies, Insecta odonata, from Museu Nacional of Brazil.
Morning or night, everyone. First of all, I would like to thank the TDWG event for the opportunity to present this work. My name is Marcos and I'm from Brazil. I am currently a PhD student in zoology at Museu Nacional in Rio de Janeiro at PPGZO postgraduate program. And I work with neotropical dragonflies, also known in science as aquatic insects of the order Odonata. Besides me, Marcos, this work is co-authored by Dr. Ângelo Pinto from Federal University of Paraná and Dr. Alcimar Carvalho from Museu Nacional of Federal University of Rio de Janeiro. So, okay, let's get started the presentation, which is titled Digitizing Primary Data on Biodiversity to Protect Natural History Collections Against Catastrophic Events. The type material of dragonflies, Insecta odonata, from Museu Nacional of Brazil. So this work is about natural history collections. Let's know a little more about them, as well as the importance for biodiversity and science. Natural history collections are guardians of biodiversity and essential to understand the natural world and its evolutionary process. They hold samples of morphological and genetic heritage of living and extinct biotas, helping to reconstruct the timeline of life over the centuries. Therefore, studies carried out in natural history collections are essential for development of the scientific knowledge and are pivotal for the scientific technological progress of a nation. Inside the natural history collections, there are deposited specimens, and these specimens have information related to their location, habitat, etc. For this information, in science, we give the name of primary data. So primary data from specimens in natural history collections are crucial elements for research in many areas of biological sciences, and also considered the bricks of systematics and therefore one of the pillars for evolutionary studies. As we have seen, primary data are essential for science, but even more important is its digitization. For this, there are projects such as Sistema de Informação sobre Biodiversidade Brasileira, known by the acronym CBBR, which digitize primary data from specimens deposited in natural history collections. The CBBR project is the Brazilian hub of the Global Biodiversity Information Facility, known by the acronym GBIF. It digitizes primary data from type specimens and provides tools for organizing, publishing, and consulting online data. In this context of natural history collections, as well as primary data, now let's know about the Museu Nacional of Brazil, the institution where the data for this work came from. The Museu Nacional is the oldest scientific institution in Brazil, founded in 1818. Known for housing one of the largest and oldest collections of insects in the world, the entomological collection of the Museu Nacional had more than 3,000 holotypes and approximately 12 million specimens, of which about 96% were lost in the tragic fire occurred on September 2nd, 2018. The CBBR project was active in that collection from 2016 to 2019 and enabled the digitization and preservation of data from the type material of many insect orders, including dragonflies. Due to the end of the agreement between CBBR project and the Museu Nacional, most of the obtained primary data are pending full curation and therefore are not yet available to the public and researchers. Inside the entomological collection of the Museu Nacional, there were some insect collections. Among them was the magnificent dragonfly collection. It was founded in 1940 by odontologist Milton Santos. This collection housed the biggest and most important collection of dragonflies among all Central and South American institutions. It assembled most of the physical records of neotropical dragonfly fauna gathered over the last 80 years, many of which are of undescribed taxa. Therefore, in order to ensure the preservation of your data and its history, this study aims to gather, analyze, and publicize primary data of the type material of dragonflies housed in the entomological collection of the Museu Nacional. As a result of this work, we have a total of 11 families, 50 genera, and 131 species. In this table, we can see the number of families, genera, and species mentioned previously. In this graph, we can see that within the suborder Anisoptera, the libellulid was the most diverse compared to the other families, with 28 species representing 47% of the total. Here are some holotypes photos of each Anisoptera family registered in the CBBR project. On the other hand, now talking about the suborder Zygoptera, in these graphs, we can see that the Cenagrionid was the most diverse compared to the other families, with 58 species representing 81% of the total. Here are some holotypes photos of each Insecta family registered in the CBBR project. The entomological collection of the Museu Nacional housed 105 holotypes of dragonflies nomina, representing 11.7% of the richness of the Brazilian odonata fauna, which has 901 species, a country with the highest number of species of the biosphere. The impact of the loss of this collection to studies of these insects is unprecedented and irreversible to biodiversity and science, since some enigmatic and monotypic genera such as Brasileogonphus, Fulminagrum, and Hopaneura lost 100% of their type series, in addition to tens of thousands of paratypes. So, in conclusion, what is the importance of digitizing the natural history collections? The answer to this question is very simple and straightforward, which is to preserve biodiversity data for the future. Since digitization projects such as CBBR register and preserve these primary data from specimens deposited in natural history collections. And the last question, how does this work contribute to science? First, it records and preserves primary data. Second, it reiterates the importance of curating and digitizing biological scientific collections. Third, it shows the extreme relevance for preserving information on biodiversity. Fourth, it provides support for future research on biodiversity. And finally, to finish, this work shows that digitizing and interconnecting digital extended specimens data is one of the main and most effective ways to protect natural history collections' heritages and their primary data against catastrophic events. Here are some references I used during this presentation. And here is the address of the published abstract of this work. My special thanks to the editors, reviewers, and all the TDWG 2021 staff, as well as the CAPS and CNPq funding institutions, and PPGZO postgraduate program from Museu Nacional and CBBR project. Here are the OrcID links for all the authors and also our email address. Please feel free to contact us. We will be happy to answer your questions. So that is it. See you next time. Bye.
Thank you. Marcus, did you make it onto the platform to answer any questions? His connection was unstable, but he is around.
Shows up in the chat. If he's
okay.
maybe into co-host, maybe if he's here. I'm not sure if that's in my buffer or I do see him.
I think I saw him come. I saw him pop up at one point. Avery, do you guys see him? Is he co-host?
Yeah, we see him. We've prompted him to unmute himself, but he has not yet. So he might not be available. He might just be in on Zoom and I'm sorry, in on Huba and not within Zoom. But we have prompted him
yep.
To unmute.
I think he has.
Yes.
So he has unmuted himself. I can see it. Thank you. Okay.
Okay. So there is two questions. One from Anna Dalmonin. How much has been digitized besides the labor data of the type specimens? Can't see him.
He's muted again.
I'm not sure. He might be having issues. I've prompted him several times to unmute, but he can always answer the questions in real time within the Whova app.
That's true. I would also like to know whether DNA data had been taken. When you have such a catastrophic event, photos are certainly much better than not having anything at all. But for many species, it's not sufficient. And certainly with the number of specimens or species that you had that were unnamed at this point. When people are doing digitization, what kind of effort is being put into getting DNA samples from various things? Anybody?
This was our last session, so everyone is welcome to unmute themselves and chat and have an open discussion if you'd like.
Oh, it's not the last session of the day, but it's the last session before a significant break.
Yeah. Our last speaker of this session.
Right. So there's what? I think the next one starts
5.
Depends on where you are. Anyway, it's SYM 14, Community Building for Our Shared Data Future, is after this significantly. You can go out if it's still light where you are, or go out and find some night critters for the iNaturalist Worldwide BioBlitz. Or daytime. Okay, somebody's in the chat. An hour and 45-minute break. Note that you can add questions and they can be answered in the Q&A at any time. So if you come back and look at this and think of a question, please add it. Make sure you address it to whomever was speaking. That will help be able to sort which speakers need to be answering which ones. So,
Any questions? Yeah, anybody? I was going to add what you said, Gail, this upcoming session, what was brought up this morning about the need for working across different disciplines and different stakeholders, right? And how do we do that? So we often have our own needs clearly in mind for our own research purposes, whatever it is we want to accomplish. But can we, and we can't always, obviously, but can we benefit from stepping back and looking at what communities are potential users of this data? Why is it being gathered? And are there things we can do to make this data relevant for other people? And I think the policy discussion was very clear this morning, right? That it's got to meet their needs if they're going to use it. So.
And are they using it without knowing whether it meets their needs? Is it always appropriate? Are they aware of the biases?
Right, that too. But feel free to open your mic there, Rebecca. Feel free to chime in.
I'm just
100% agreeing with you as well, because that's something that we talk about in our session as well, is the importance of expanding the disciplines that we build our infrastructure for, but also who we build our tools for, so that it's not just the narrow niche that we are used to focusing on. So yes, plus 100 is all I was saying.
Yeah, I'm not sure in terms of who's in the audience, because there's like 100 of us here. And your question, Gail, so is not sure that people here that are doing digitization, or these are the people who are supporting standards development and tool development. So James, you're here from the plant direction. Are y'all doing DNA extraction with digitization? Put you on the spot if you're still here. James Macklin?
Hold on.
There you are. I heard your voice for half a second.
Yeah, I'm here. I don't know about my camera though.
No, well, don't worry.
All right. So yes, in some cases we are doing targeted extraction on things that we digitize, so that is part of a program project that we have. I mean, I think more what we're calling molecular characterization is becoming more common as that technology gets easier to do, and of course as we have more need for it. So I mean, this is another part of that linking story. Mm-hmm. But you know, again, it comes to resources, right? So having the resources to do both the digitization side and including molecular sequencing as well as a service, well, you know, you have to have a fairly good want to do that. And usually those things are project driven, not necessarily just like we do in digitization sometimes of start at A, you know, and move to Z. So, you know, I guess at some point the technology will become so germane, like imaging has become, that we'll have a lot more sequences. But to this point, fairly project driven.
So as Marcus and Gail pointed out, though, even at that point, it's like, it's not just about the science question that you're trying to answer at the moment. It's about insurance for the future. If those specimens were to be lost, destroyed, or otherwise gone, the more evidence you have left behind, the better. Yes, so it becomes, as it becomes cheaper, it also it may be more needful too.
Mm-hmm. I mean, is that something that the funding agencies, such as they are around the world, are aware of, that with the digitization effort, that taking DNA samples from either type specimens or paratypes at the very least are things of great importance?
Patricia has her hand raised. Patricia?
Can you hear me?
Yes.
I'm on my tablet. Oh, okay. So I remember in Museu when we were doing mass digitization, we tested and envisaged to take DNA at the same time, but it would slow down the workflow too much, because we were with Picture to do the mass digitization, and it would make it too expensive and take too much time per imaging if we would stop it and take DNA. So I know they do more targeted sampling when there is a project or research, but it's not done necessarily at the same time as the mass digitization. But if you do targeted digitization manually, then yes, you can take DNA at the same time. But it really depends on your digitization workflow. And those with the conveyor belt, it's not impossible, but you have to count that you will take too much time to process all your specimens as you have the objectives.
Handy Bentley put a comment in the chat. Handy, I don't know if you would like to open your mic. So comment is, it's a cost per unit effort question too. How much more time would it take to digitize a specimen if you had to sequence it? And how many specimens would go undigitized because of that? Deb?
I think, yeah, that this balance is always important, but I would frame it a different way. When we're saying digitization, can we separate the difference between the huge backlog that we have and the need to keep that backlog from getting bigger? And so thinking about what does this mean for specimen collecting that will happen in the future? And there are many issues with future specimen collecting, permits and Nagoya and access and benefit sharing, et cetera. But I think in the future, right, the specimen has a tendency to probably come into the collection with its sequence, or at least some part of it sequenced as a matter of the way in which future collecting will be done. Yes? Thoughts?
I know like I know on like the research people who are doing like genetics, there is like one project was like the Earth Genome Project, where they want to sequence every species or at least every family on Earth. But I don't know if like those groups who are involved in that effort has any connection to the people in the collections. So I know there are wishes to do this, like with, you know, things and talks and discussions, but I don't know if any of the people in the museum collections are involved in those discussions.
Good point. Can you say hi? I'm not sure who that was speaking. I'm sorry.
Hi, Yin Kwan.
Hello.
Hello. And I know those groups have tried to get funding, like from some of the big benefactors, because this is like a multi-million-dollar effort. You know, it'll take millions of dollars to do this. So you might try to connect with those groups and start a discussion of trying to get collections involved.
Could you put the names of those in the Hoover, in the chat inside Hoover? That would be great.
Deb, some of that initiative is led by the Smithsonian. Does anybody from the Smithsonian can comment about that?
I am actually at the Sanger Institute. We are carrying on the Darwin Tree of Life project on site. And at least for this project, there is a sampling working group. So there is a tight association, a communication between the scientific collections with from the beginning till the end of the process. So from the people they are collecting to the people they are registering, separating the tissues that will be used for specific, how do I say, sequencing strategies up to the point where an assembly, a genome assembly is released. So yes, in this case, there is a tight communication between the people they are sampling, the people they are storing, and the people they are sequencing. I don't know if that helps.
Patricia has her hand up.
Yeah. Yeah, but maybe, you know, at the European level, there is a European call that was for submission now in October, that is called barcoding, I think, life on Earth. And actually, it's the museum community, natural history, that are even leading it, and that are some of the main partners, together with, of course, also other stakeholders from more the environmental point of view. But for sure, in Europe, there is a strong connection between the barcoding of life on Earth and the natural history institutions, for sure. So that's really guaranteed. And they also work together with the US and Australian groups. So there is surely a strong connection with everything that is barcoding and the collections and the museums.
Are there any other comments? Things that people would like to talk about?
So I'm at Denver Botanic Gardens, and we're pretty small natural history collections, but we've started collecting tissue samples with every voucher that we then silica dry in hopes that someday we do sequence.
Oops.
Well.
He was just pinned and
Yep.
we lost him.
You know, he's rejoined already, but you got the relevant part out. Yay, Rick. So for all of you that's Rick Levy from Denver Botanic Gardens.
All right, I'm back. Sorry.
Yay, you're back. It
just went black. But so yeah, we're collecting tissue samples along with every voucher so that there's a genomic quality sample associated with every voucher in 99% of the cases. But I know that we're doing this part of the GGI and GGI Gardens project, which I think is shutting down this year, but is kind of evolving into this or folding into this. I'm not really sure. Earth Biogenome Project. But I did a while back, I read a pretty good paper about, and I can't remember who the authors were, but it was a whole workflow for sampling herbarium specimens for genomic data, and they had a great workflow on like kind of how to, I guess, go through it in a pretty standardized methodology. So I know that I'm not saying it's not necessarily part of the digitization, at least here, but like what Deb is saying, certainly in the future, maybe someday we come back to the collection already with sequence in hand, and that'd be pretty slick.
Anyone else? Do we have another question? No. Let's see. There's.
I have lots of questions, y'all, but it would definitely might go off on another tangent.
Go ahead.
I was curious about an earlier topic that came up about re-imaging, and I think it was in the prior symposium. So many people here are familiar with we're capturing data, right? Digitized in a variety of formats. And the two things, I don't know which ones y'all might want to talk about, but one was this notion of re-imaging, because imaging technology changes. And I know that if Barbara Tears were here, she'd tell you the story. This is not my story, but hers, of her epiphany after the second time she took pictures of the types at the New York Botanic Gardens, recognizing that it wasn't going to be the last time. And indeed, last time she gave this talk, it's now been done 3 times. Because the imaging technology changes so much, the quality of the images and what you can do with those images from a research standpoint has changed immensely. So thinking about that in terms of DNA access and what does that mean to our community for capturing information. And the other part I'm interested in came up more or less because of COVID. Some of you may know the acronym IoT, or Internet of Things. And the notion of when COVID happened, all of a sudden digitization was something that, at least if you had labels, it could be done from home, it could be done from your desktop, you didn't have to be in the collection. But you couldn't monitor for pests, for example, in your collection. Nobody could go into the building. And so at least that the AMNH, the American Museum of Natural History, began to experiment with IoT. So they, using cameras, could, for example, they did a very manual thing. They had a a fan vent for like humidity control and all this, and they want to know if the fan is working. And so they put a little string tie on the blower so they could see if it was working, and then they put a camera on that so they could watch it. But that way remotely they could keep track to see if that was working. And there are other ideas out there, but they were trying to implement, you know, can we see if there's anything in a trap, for example? Can we monitor our freezers? Are they working? Are they okay? And then I was thinking about pest management, and if we're ready yet to start, I know a couple people have reached out since spinach, but you know, nothing should be moving inside the drawers. They're supposed to be dead. So putting sensors in place so that you could manage pest problems. I noticed somebody the other day said they only check the collection once a year. It's like a lot could be eaten in a year. Anyway, thoughts about Internet of Things in any of this, or thoughts about imaging? And thanks for sharing the paper, Rick. I see it in the chat.
Left to wonder, in what regard does imaging equipment change? Is it just resolution, or is it also like truthfulness of the colors? Or I can't imagine it's color depth, because usually you'll stick with your 8 bits per channel, but like in what kind of, what area are those improvements, those vast improvements?
Well, at least speed and resolution. I mean, the Global Plants Initiative had a 600 DPI, but the scanner took like 4 minutes to run one specimen.
All right, that is a long time.
That is a long time, yeah. What was cool about that project, though, is the scanners were well built and very industrial, very sturdy. And after the project was over and they were still operating, they were able to share them with other collections that had not initially been part of that initiative. And it was originally the Latin, somebody here must know more about it than I do, but it was originally for Latin America, and they were able to broaden it to the entire herbarium community around the world. But so while it was slow, it was robust. Well, I'm seeing Jim's comment in the, yeah, it is ironic, James, but yeah, so if you have the time, the question is, how do you evaluate that? Do you have time?
Patricia has her hand.
Yeah, well, on the topic of reimaging, sometimes it's also for very practical reasons, because if you do mass digitization, and sometimes it takes more time to go take out the herbarium sheets, for example, that have already been digitized, and to take them out, and rather take the whole bundle and redo them, because it's a mass digitization workflow. So it would take too much time to go check, has this one already been digitized, and taking it out. So there are sometimes quite a few that are re-imaged just for practical reason. And when I talk to my colleagues who do more, I would say 3D digitization or CT scanning, there the technologies evolved really a lot, and they also test different devices, and they evolve quite quickly. And so there they do sometimes quite a lot of re-imaging of the same specimens to have a better quality, or to be able to extract more features from this 3D scanning. So I think re-imaging is more done when you do 3D and CT scanning than maybe when you do 2D scanning, when I talk to the specialists.
Anything else? I'd like to thank our speakers, both virtual and, well, everybody's virtual, pre-recorded and in person. And please make sure that you revisit pieces of this session and add to the conversation, and encourage people that may not have, who may have missed parts, to come back. Thank you very much. Let's see. And as we mentioned before, the last session of today is SYM 14, Community Building for Our Shared Data Future. And that's in a little over an hour, I think. So take a break. Thank you, everyone.
On the agenda, it says something of a social event in half an